you doing this morning, Mike?
Oh, I'm doing well. How are you?
Great. Yeah, living the dream.
Yeah? Ready to get some Rivercane?
Heck yeah.
Could run upstairs and get our other stuff, but three samples of what I would call full or old leaf. We want to do 3 young leaves. We'll look for more inflorescences while we're doing it. We probably want to maybe we can get some culm.
Okay.
Like if there's some culm that you feel like we could cut off, and then
Okay, you can cut.
okay, and then some we'd want to get some rhizome and then some separate root as well.
Okay.
We just kind of want to run the gamut on
cool.
everything, and because every little piece does things a little differently,
Okay.
and so that's information that we could use. And so, from a perspective of like, as a person who does this science, like for science, what we'll try to do is, I could see us wanting to sequence the genome, annotate it, so find all the genes using all this information, and then we'll push it out there as a resource, and then we can start building our genetic diversity stuff and asking questions about, here's all this cane from these different areas. Like, for example, one thing that Mike and I have talked a lot about is doing flooding tolerance experiments, because I've heard some people say it can't handle being flooded for a long time, but then there are breaks that seem to be fine with that. So let's ask the question, do they? How is it doing it? What's different? So there's just a lot of really fun things to do, and there's a great opportunity for students to learn stuff, and I'm hoping, like, Mike and I have talked about this, I'm hoping that we get to have like some of y'all come to the lab.
Oh yeah, love to.
And do this kind of stuff, and there's even, if people are ever interested in doing the computer-based part too, like if you guys have students or if you want to learn it, whatever, we can do that remotely too, and I can get you guys access to our computer, our big cluster at UA, and we can just work on it together and just do some cool stuff.
I'm all for that.
Sweet. Yeah.
Stalk of the Rivercane from the ground up. That's a culm, and then the node is where the branches occur off of the node, and that's where the node is right here. And this in between the nodes is called the internode, and then the branching's off. The more branches the Rivercane has, the older it is. The younger Rivercane only have 1 or 2 branches on each node. So what are you what do you collect out is for the seed thing?
So what we're gonna do is we want to collect different of these. We call them inflorescences, right? It's all where all the little flowers are, the little florets are. We want to collect them at kind of different stages. So like if you're like, oh, this one's got a seed in it, so we want to collect that one, or we want to collect it before the seed. We'd like to collect the one that has like the pistils. We want to just and if we can find any that are even smaller and younger, like coming out, just because what happens is that different genes get turned on at different times, and so we're interested in saying, oh, well, what do they do when they want to make a pistil? What do they do when they want to make a seed? And the way that a lot of plants develop is they, especially grasses, deep down inside of like their leaf sheets, they kind of build the flowers, and then they expand them. So some of the really good genes are when they're tiny. Cool. So there's a lot of cool stuff going on here. So this is a young inflorescence. It hasn't extended much yet, and it's still the florets, which are what we call the flowers because they're little flowers. They're probably fully developed, or they're close to being developed, my guess. But but still a good time to catch it because they haven't fully developed like their anthers yet, so they haven't released pollen or anything like that. So it gives us a new point of view of like what genes are involved in making flowers.
If we would have got that one down there, that'd been perfect, because we were just sitting there eating lunch, and I noticed this turn purple. I was like, that's flowering right there.
The purple stuff would be really interesting because so with plants when they turn purple, so they have chlorophyll that makes them green, but then they have other pigments all underneath the chlorophyll, kind of. So some plants, they turn purple when they're exposed to sunlight all of a sudden. Like, if it's a response. They also do it when they get cold sometimes. But I'm wondering if you said that they start to die afterward, it might be that they're breaking down their chlorophyll and using it for like energy, and then that's what's left over is the purple, which would be kind of cool, and it's a really interesting signal to say, oh look what this plant is doing here. It's really neat. I think there's a cool physiology thing going on with the plant. So just gonna make sure this is tight before we drop it. So we'll drop it in, and so there's like there's a little distance, and then this has got a nice jacket on it.
Yeah.
So you can freezes it real quick.
So is it like a pool of freezing stuff? Or?
Yeah, this is a 10-liter container of liquid nitrogen. So it's been evaporating since yesterday, but you can hear it. It's bouncing.
Yeah.
So there's still plenty in there, which is good. So we've got a plan to, like, in case it evaporates out, because like keeping it closed helps out a lot. But you can see it coming out now. We'll get some dry ice and leave them on dry ice. But this flash freezing, this helps get it, takes care of everything. So yeah. You want to do some dropping?
No, go ahead.
Are you sure?
Yeah. I'll make sure the camera doesn't fall out.
Okay.
That's cool.
These are inflorescences. These are, just wait a second here.